Good evening, everybody. Welcome to Turfgrass Epistemology. My name is TravisShaddox. Thanks so much for coming here tonight. Sorry if I can speak. I was trying to get some last minute things put on here, and I don't know if I can get them or not, but I'm going to do my best. This is Turfgrass Epistemology. If you're here, you're wondering, how do we know what we know about turfgrass science? We explore the scientific literature. We go over videos sometimes. We're going to go over a very short video tonight. And then we're going to go over a scientific article tonight. And then I'm going to be off for a couple of weeks. So I'm sorry for those people who look forward to seeing this every week, but I'm going to be gone for a couple of weeks. It's spring break here in Lexington, Kentucky. So I'll be gone with my family. And then I'll be back for the foreseeable future on a regular weekly basis after this little break. I had a lot of good comments on the members only show yesterday. I went over a bunch of fertilizer stuff, which, to be honest, I thought was kind of boring, but I showed how fertilizer particles are measured and distributed and SGN sizes and things like that. And apparently people like that. So I appreciate the people that came and watched that show. If you want to watch those shows on Monday morning, you'll need to be a member. You can listen to it live on a podcast, but yesterday's podcast that got released this morning, I don't know how useful it's going to be because there's a lot of visuals in it because I was showing things on the video. But that's what you get for being a member. You get extra stuff like that. So tonight we're going to, I think what I'll do tonight is wrap up the soil health topic. I started in January after the Christmas break going over soil health. And I thought I would get through it by now, but I've barely even started. And it's April. So at some point, I think I've sort of need to maybe change subjects at some point. So I'm going to go ahead and switch subjects probably when I come back from the break. But I'll have to come back to the soil health at some point because I haven't even got onto the Haney tests or any of the other wishful thinking tests. We've only been talking about the Solvita test for the last couple of weeks. And we talked about the Solvita test a week ago on the members only show. And we're going to discuss it briefly here tonight and then show what the results are from a research team that does not have a vested interest in the outcome of the project. So that's what's going on. That's kind of what's on my mind. I'll probably come back and start going over a lot of water, irrigation, and things like that, I suppose, is probably what's going to happen. Okay, so as I get started tonight, I wanted to first talk about the first comment in the chat. And I'm mentioning this only because this is my fear, is that people are going to see this and think, oh, I should go out and do this test. And the comment is from Brandon Clifford. He says, this seems like quite the topic. Never even knew this was a thing, being a soil Solvita test and relating it to nitrogen and the needs of nitrogen. It's not a thing in turfgrass. Let me be absolutely clear, okay? Don't run out and do the reason I'm showing these tests and going over these tests right now is to cut it off before it gets started. Okay, there may be ample evidence a year from now, a day from now, tomorrow there may be a whole flood of evidence come out, or maybe 10 years from now. At some point, we find that these tests are very useful for turfgrass management, but not now, not on April 1st, 2025. So please do not go out and buy these tests and start using these Solvita tests and the Haney test and the soil health test and start running all these things thinking you're going to develop a more efficient turf management program or nutrient program, because it's probably not going to happen. We're not there yet. I don't want to stifle progression. I don't want to slow it down. I'm glad these people are going out and doing this work and working on it and keep working out all the problems. But there's so many problems with this system, being soil health and carbon dioxide burst technology and labile ammonium nitrogen technology and all these other tests that they're doing. There's so much variability and so many problems with them. And there's essentially no correlation between those data and turfgrass response to applied nitrogen, that it's not useful right now at all. So please don't go starting to use these tests and then starting to fidget with your nitrogen applications up or down. Because if you're following best management practices and you're applying nitrogen within best management practice ranges, and more specifically, if you've adjusted your nitrogen rate up a little bit to meet your nutrient or your turfgrass response needs, or adjusted it down a little bit, and you find that you fine-tuned the amount of nitrogen that you need, then you don't need any of these tests, even if they were 100% accurate, because this test is going to measure the amount of labile nitrogen in the soil and then provide some prediction as to how much nitrogen you should apply. But if you fine-tune the amount of N that you need, that does include not only the nitrogen that you apply, but also what was already soluble in the soil or being mineralized from organic matter in the soil. So you don't need this stuff if you've already calibrated your nitrogen at your location. Okay. So thanks for the comment, Brandon, because this is my fear, is that people are going to see this and go, oh, well, I didn't know it was a thing. Let's go do it, Don't go do it. Okay. And the day they present and publish enough evidence that's convincing to me and compelling to me that we should start using it, then I'll be right here talking all about, let's start using it. But until then, do not go run out and start using these tests. The same thing goes with the ion exchange membrane test, as you all probably know. The senior viewers of the show already know that. Don't go start using the ion exchange membrane test and the Solvita test and the Haney test and all the MySoil tests and all. Don't do that. A standard extraction method, Malick or Bray or Olson or whatever is appropriate for your soils and your location, is useful for everything except nitrogen. There's not a test right now that we can use to help manage nitrogen applications in turf grasses. Not yet. I mean, there's not a good one. There's not a valid one. Okay. With that, let's get started. Now, I'm going to first show this short little video. If I can figure out how to get this. Okay. I think that's it. Okay. So on the screen is a video from a YouTube channel called Woods In Laboratories. The title is Solvita Soil Carbon Dioxide Respiration Update Protocol Improvement. And this is Will Brenton. You're going to see his name once or twice tonight. And it says he's the president of Woods In Soil Labs and the inventor of Solvita. I'm going to skip forward. He just mentions a few little introduction things. And then I'm going to skip forward to actually how they performed the test, because it's important to understand how this test it functions, because the paper we're going to go over tonight is very, very similar in the methodology, except how they actually measure the CO2 in the chamber itself is different. And I want to show a little bit about that if you're not familiar with it. Here we go.
To perform the Solvita CO2 burst test, take a soil sample that has been oven dried at 35 to 40 degrees Celsius and then rolled. Scoop a subsample with the 30 cc scoop. Take the striker and level off the subsample in the 30 cc scoop. Then take the beaker and place the subsample inside the beaker. At this point, you will weigh the beaker to determine the soil's bulk density. Then take the small mesh screen, place it on top of the soil sample inside the beaker. 9 cc's of water is then added to the beaker. The screen is there to help the water disperse evenly and gently. Once the water has filtered in past the screen, take the screen off the soil subsample, place the beaker inside the jar.
Okay, so up to this point, everything you're going to read tonight on the article is essentially the exact same as what she's done here. Okay? There's nothing different for the most part. What she's done as compared to what we're going to see later in the episode, in the article tonight. Okay?
Then open the pouch. Take the paddle from inside the pouch, place it inside the sample in the beaker. Take the cap for the jar and twist it tightly onto the jar.
Okay, so right now what this is just so those people who may not be familiar with this process, she's put some moisture into a cup with a known volume, the known volume of the solution in the cup. a little, probably 100 mil or 150 mil beaker, and she put that inside a jar and sealed it. And she has a little paddle, a colored paddle in the soil. What's going to happen is the microbes in the soil are going to start breathing, essentially, and the gel on this blue paddle is going to start to change color based upon the concentration of CO2 that remains in this chamber. Okay, that's the basic concept. All right. I have zero problem with any of that. The idea, the concepts, I'm okay with. But the red herring that exists in soil tests like this is that this what's going to happen is this paddle is going to change color, and then they're going to read that color change, and then that's calibrated to a carbon dioxide concentration. Okay, fine. The red herring is I don't care. They're going to say that concentration is relatable to mineralizable nitrogen or available nitrogen, and based upon the concentration, you should either apply more nitrogen or less nitrogen based upon the concentration of carbon dioxide, or the respiration of the microbes in the soil. All of that may be true. What I'm saying is we don't have enough confidence in providing that prediction. Even their own paper doesn't provide sufficient confidence in predicting how much nitrogen we should be applying, although their paper will say they do. It doesn't. Okay. Now, the paper tonight we're going to read, those are the papers I went over on the members only channel. The paper tonight we're going to read is essentially this exact same setup, except they're going to use an instrument that is far more precise and far more accurate than this paddle. Okay? Just so we're on the same page. We're going to finish this off in another couple, maybe 30 seconds.
The jar then goes into an incubator for 24 hours. After 24 hours,
I screwed it up. What did I do? Is it playing?
The jar is removed from the incubator, the cover removed, the paddle is removed from the beaker. With the DCR in the low CO2 mode, place the paddle inside the DCR.
Okay, so she puts that paddle inside an instrument that reads the reflection of light off that paddle and gives a digital readout of the color of the paddle, and then they calibrated that to carbon dioxide, okay? That's called the carbon dioxide burst test, where the soil was dry, they wet it, and then there's a sudden flush of microbial activity, and that carbon dioxide flush off there is called carbon dioxide burst. No problem. And I have no problem researching this. Let's do it. But the problem I have is when you're going to go tell a turf manager that you should or should not apply X amount of N because of that, when there's zero, I shouldn't say zero, there's essentially no evidence to support such a recommendation. There's 2 papers on this, maybe 3-ish. There's 2, well, on this particular technology, the Solvita thing. And both of them conclude that it can be used. And in both of them, their data doesn't support that conclusion. Now, what I mean by that is this. Now, let's pull up their papers. First paper I went over on, oops, that's not the paper I want to go over right now. The first paper I went over on the members only channel, or the members only show, on Monday, a week ago, I think it was. I'm not going to do this whole paper. I'm just going to go down here and read the conclusions. The conclusion was, where is it? The conclusion was this: the Solvita Layball ammonium nitrogen test should be able to detect turfgrass soils with high nitrogen mineralization potentials that would allow for reduced rates of nitrogen fertilizer applications without sacrificing turfgrass performance, as well as identify low mineralization sites. This is going to be critical to tonight's episode, okay? Low mineralization sites that justify higher nitrogen inputs. What would be a low mineralization site? Something like a putting green, okay, or pure sand root zones, or soils that have less than, say, 1 or 2% organic matter. Something like that would be a low mineralization potential soil, okay? That was with labile amino nitrogen, SLAN, okay? The other paper that they published, oh, by the way, I wanted to point out before I go to the next paper, is that the authors are, right here, William Brenton, okay? And they state down here that the conflict of interest, here's the conflict of interest on the paper. They make a conflict of interest that he's the owner and Carl Gillard gets some free stuff from the company. And both of these guys, William Brenton and then all these authors, here's Carl Gillard, he's emeritus from Connecticut, I believe. And so, in other words, what I'm getting at is the authors have a vested interest in the outcome, regardless of what they say, okay? They have a vested interest in publishing evidence that supports buying this product, okay? Doesn't mean the data is wrong. Doesn't mean the paper's flawed, although I believe it is. Doesn't necessarily mean that, okay? The data could be 100% accurate. I'm just saying you have to read this with a great deal of skepticism when you know the authors have a vested interest in the outcome. Now, the next paper they published, which is part 2 of that, you notice this, it says right here, part 2. I'm going to go over this probably in, I don't know, the fall or something. I was going to go over it now or this season, but I'm getting tired of going over soil health stuff, to be frank. And this is carbon dioxide burst. Okay, this is what the test is they just did. And again, I'm just going to read the conclusions. And you'll notice it's the same authors, William Brenton and Carl Gillard and David Moore. And so we know they have an interest in the outcome being positive. Okay. We also know that doesn't necessarily mean they did anything wrong. It doesn't necessarily mean the data is inaccurate. It could be all correct. Just read it with skepticism when you have a situation like that. And in fact, on the members only show on Monday, I was reading that paper and I didn't even know that they had a vested interest in the outcome. And I was reading the paper and things weren't lining up. They were making conclusions saying you should do this. In fact, let me show you. You should do this because there's significant relationships. And meanwhile, when you see the relationship, the R squared is 0.12, 0.19, 0.04, 0.08, 0.07, 0.18. Yeah, they're significant, yes, meaning there's a probability of there's a less than 0.01% chance that they're wrong, meaning there is a relationship there. But the relationship is so variable. No reputable scientist would ever say that they can provide any meaningful confidence in a predictive ability of the test because there's so much variation. Okay. And now when we go to the carbon dioxide study, the conclusions say this study suggests that cool season turfgrass lawn responses are positively correlated with this Solvita carbon dioxide burst. Concentrations in turfgrass soils can be categorized by their probability of equaling or exceeding urea benchmark values based upon these concentrations. Relatively high variability in the test, however, will introduce more uncertainty in the prediction outcome. So they make a point. They say, they don't necessarily hide it. Relatively high variability. The variability is so high. How can you, with any conscience, provide any confidence that your prediction is going to be accurate when the R squared is 0.1 or 0.04? I think the highest R squared was 0.3. So, anyway, so the last sentence in the conclusion was the carbon dioxide burst test can detect turfgrass soils with high biological activity, and this should allow for reduced rates of nitrogen fertilization without losses in turfgrass performance. Low mineralization sites that justify higher inputs can also be detected. Again, things like putting greens, sand-based root zones. So they're making conclusions in a refereed paper based upon basically what is this. I'll show you again on this paper too. R squared. So I'm looking at the CO2 concentration on Kentucky bluegrass, tall fescue, or both species. We're looking at NDVI, chlorophyll index, yield, all these things, okay? Nitrogen uptake, all these things. And the R squared again is 0.2, is a little higher, 0.2, 0.3, 0.2, 0.3, 0.1, 0.05, 0.16, 0.9, and 0.1. I mean, they're so low, it's laughable. Essentially, what it's saying is, yes, there's a relationship there because the p-value is less than 0.001. But I would have 0, or I would have 0.13%, or I would have 13% confidence, essentially, that I could give you a recommendation and actually have that recommendation result in a greater outcome in turfgrass than if you didn't follow it. I would never do that. I would never do that. I would have to have that R squared up like 0.5 or 0.6, really. And even in the field, and I think they even did this I think they pulled them from the field and they did it in the lab. But you need to get these R squareds up higher if you're going to have predictive abilities. Like I just actually was sending a manuscript to a co-author yesterday or today it was, I can't remember. And the R squareds in there in some cases were low, very low, like 0.01. In other cases, during other seasons, it was 0.6, 0.5, 0.6, 0.7. So a normal, I think, a reputable author would say, this study can be used to predict outcomes in these seasons, but in other seasons, it may not be reliable. We can't use it. In this particular study I was working with Malic 3 sulfur. So in other words, when the soil sulfur was low, you can use it to predict outcomes. But when the soil sulfur was high, there was no response. So I can't really predict an outcome. You have to interpret the results within the boundaries of the results. You can't move out. And that's what these authors did. Now, I don't know how this happened. My cynicism and my joy in seeing con artists called out tells me I want to say this was intentionally done, but knowing the authors, it probably wasn't intentionally done. It was probably just out of interest in science and interest in knowing what actually happens. But it just so happens that another group of scientists, in this case, they don't have any vested interest in the outcome, published a paper in 2023, just a couple years ago. And this is entitled Short-Term Soil Carbon Mineralization on Golf Course Sand-Based Putting Greens and Its Effect on Creeping Bentgrass Nitrogen Uptake. Essentially, this is the exact same type of setup, but they're going to be looking at putting greens, and instead of using this little paddle, they're going to be using an actual instrument that is much more accurate and precise. Okay? So before I jump into that, let me read if there's any comments real quick. John Carman says, isn't CO2 a nutrient for plants? Yeah, carbon is the highest nutrient for plants. Carbon, hydrogen, and oxygen are like 95% of most plants. Carbon is usually, I can't remember now, it's usually like 50 or 60%. Or no, actually, I think it's more like 40 or 50%, and then oxygen is some other higher percent or some other percentage. I can't remember all the percentages, but yes. But the plant gets all of its carbon from the air. There's no carbon that the plant gets from the soil. So as long as it's breathing, it takes in carbon dioxide and releases oxygen. That's how it acquires carbon. Yeah. So carbon dioxide I wouldn't say it's a nutrient, but I mean, carbon is an element, a plant essential element, but you wouldn't apply carbon dioxide as a nutrient. It's not in that regard, no. Andrew Winters says, welcome to Entry Level, a new member. Thank you very much, Andrew. I appreciate it. I saw Chuck donated, or was it Chuck or Joe or somebody donated something, a membership. I'm not sure what happened earlier, but thank you. Mike Conroy says, will fungicide applications alter the test if you killed off some microbes? I'm not a soil microbiologist, Mike, as you know. But from the little bit, don't take what I'm saying as truth, okay? Because I'm not a soil microbiologist, but I do listen to some microbiology talks here and there when I go to these scientific meetings, and I do believe that is a consideration. Soil microbiologists do account for that or consider that if they're going to be measuring that. They want to know if there's been anything applied that would have reduced the populations or their activity. And I believe the answer is yes. Okay, but I'm not a microbiologist. Well, yeah, this is the question. This is the question you should ask, Mike. If the paper is refereed, how did it make it to publication past all the referees? That's a good question. As you've seen, Mike, on this channel, you can put up a number. I don't know what the number is. I'm going to say 5. There's been at least 5 papers I've gone over that I've just hammered that are refereed from good authors in some cases. I've just tore up, and because I think they're junk. Okay, I think the authors either have a bias, or they have a vested interest in the outcome, or they're just incompetent, or they just had a bad day. I don't know what it was, but I've hammered a lot of them. But the referee process is not foolproof. It's just the best system we currently have to filter out misinformation, disinformation, errors, honest mistakes. It's just the best. It's just currently the best system we have. That's the reason I rely so heavily upon it. But it doesn't mean it's foolproof or flawless. Okay, that's for sure. Oh yeah, now my thing's working. I was having problems earlier. I couldn't get it to move up. So that's the questions for now. Okay, let me get into the paper then. Any more questions? Put them in the chat, and I'm getting better at addressing them as I go here. All right, so the paper tonight is relatively short. I'll get through it. I should be done by in the next 30 minutes, I think. This paper, I don't know how to pronounce this gentleman or woman's name. I'm sorry, but I'm going to say it's Zhou. I'm not real sure. It's a Chinese sounding name. And then, of course, Dr. Soldat, who's really at the top of the list in terms of soil nutritionists in the world, in my opinion. And then Matthew Roark. I don't know Matthew. It says Department of Soil Science at University of Wisconsin. Okay, so we're going to read this. It's called Short-Term Soil Carbon Mineralization on Golf Course Sand-Based Putting Greens and its Effect on Creeping Bentgrass Nitrogen Uptake. Nitrogen is typically the most limiting nutrient for turfgrass, and in supply is well correlated with turfgrass growth rate. See, if I have one, he's citing Wendy's paper here, 2016. There's more current data than this. So for my friends in publishing, there's a more current paper than this published in 2000 or 2001. Anyway, sand-based putting greens receive the most nitrogen per unit area compared with other components of a golf course. And again, he cites Wendy's paper again. There's more current information. And the quality of this source, the quality of the surface influences golfers' perception of the entire golf course's quality and value. UF golf courses use 55,333 megagrams of nitrogen fertilizer annually. It's a different number now because there's been updated data from this Glartner 2016 paper. So it's not exactly that now, but these nitrogen inputs pose potential significant non-point source pollution risks. Optimizing the nitrogen application rate is one of the most effective ways to improve turfgrass management and reduce its potentially negative environmental impacts. Unlike agronomic crops, maximizing turfgrass growth is not the priority of turfgrass management. That is true with everything except sport turf and sod production. In sport turf and sod production, it's almost always turfgrass growth. Grow it faster, quicker, recover, cut it, sell it, get it out. That's sod production. And in sport turf, you're in a never-ending cycle of establishment. You're never fully established. So even if you were fully established, you're going to have a game on it, and then it's going to be beat all to heck, and then you're going to have to reestablish it. So in those cases, you're looking at maximum growth. In lawn care, and you could even make the same case in, say, driving range tees or just regular tee boxes. You're looking for it to grow in quickly. But in most golf situations and in almost every lawn care situation, that's correct. You're not worried about growth. You're worried about just quality or performance or aesthetic value. Instead, turfgrass is usually managed at a relatively low growth rate to maintain turfgrass playability and reduce labor associated with mowing, but still sustaining enough growth to overcome stresses. Optimizing nitrogen supply on putting greens is important. However, modern fertilization recommendations for golf courses or for golf course turf managers are still based on empirical observations of turfgrass response to fertilizer applied on research plots, but rarely include soil analysis as a component of potential in-supply. 1 second. I don't know what it is about my voice. I start talking and it starts going. Now, I will say what they're saying there is correct, but what they're saying is nitrogen recommendations are based upon nitrogen calibration studies that we do on research plots. That's true, but we provide a range in almost every case. A range is oftentimes accounts for the aesthetic expectations that you have. That's going to differ from homeowner to homeowner, from golf course to golf course. It's also going to account for seasonal variations and geographic locations. If you're north or south in Florida, or if you're in northern Alabama or southern Alabama, or if you're in western Kentucky or eastern Kentucky, it's going to account for that. There needs to be a range because I don't know where exactly they're going to be reading those recommendations. So if you get that range and you just run with the same pounds every year, I think you might find your program to be a little more efficient and effective and more evidence-based if you just fidget with that number a little bit up or a little bit down. Move the number up or down to where you find that exact nitrogen amount that you need that would result in the desired outcome. If you do that, then this paragraph that Doug wrote, although it's true, you would be better. You'd be more evidence-based because you wouldn't be basing the nitrogen application rates off of research plot calibration. You would be basing it upon site source calibration, your own site calibration. And as long as you're doing that, it's going to be extremely difficult for someone like me to come in and argue against it. If you say, Travis, I started off, the range is 1 to 3 pounds. I started off at 2 pounds a year. It wasn't quite enough for 70% of my customers. My customer lawns weren't quite good enough, or the fairways weren't growing quite enough. So I moved it up to 2.25 pounds or 2.5 pounds, whatever. Or the other way, it's been looking great for 2 or 3 years. So I moved it down to 1.5 pounds and it still looks fine. So you're self-calibrating. Okay. So if that's the case, you started with research calibration plots. That's where we give you the ranges to start with. And then you've moved it up or down based upon site specific calibration. That's what you need to do. Okay. Okay. And if you do that, then all these nitrogen soil tests are completely irrelevant and useless to you because you've figured out what you need in your location. Okay. Let me get through the introduction. I'll answer some questions. There are two primary end sources driving turfgrass growth on sand-based putting greens: net mineralization from soil organic matter and nitrogen fertilizer inputs. That is true. Yeah, there's other inputs, but those are the two big ones. Several laboratory and in situ tests have been investigated to quantify soil potential nitrogen mineralization, but these methods usually require several weeks for incubation and are not offered in routine soil testing. A rapid soil testing method that is capable of estimating soil nitrogen mineralization would help turfgrass managers in timely making nitrogen application decisions, especially on putting greens. Studies over the past 2 decades have shown a positive correlation between mineralizable carbon and net mineralization, which was either measured under field conditions or laboratory incubations across diverse soils. Moreover, previous studies have shown a relationship between mineralizable carbon and plant performance in agricultural and grassland systems. And they have some citations here, okay? And this more here is, of course, what I just showed you. I don't see the other one. Anyway, there's another one or two in there too, but I don't see them. Okay, specifically, Franzlubers. I'm sorry, it sounds German. Franzlubers and Haney. Oh, here's the Haney test. So this is another test I'm not going over right now, but please don't be delusional. Again, the Haney test, somebody might be watching this 10 years from now or 10 days from now and going, yep, Dr. Shaddox is dead wrong. Here's the evidence. That could be the case. But as I sit here on April 1st, 2025, there is not sufficient evidence to warrant using the Haney or the H3A test on turfgrass soils to manage nutrients. There's not enough evidence to support it. So don't go down those roads. And between the Haney test and the Solvita test, that comprises a large portion of all these soil health tests that you send into the labs for. Okay. Specifically, these two authors found a strong and positive correlation between net soil nitrogen mineralization and mineralizable carbon, where mineralizable carbon quantified by a 1-day incubation. Recently, studies have used mineralizable carbon test kits on Kentucky bluegrass and tall fescue lawns and indicated that mineralizable carbon could guide nitrogen applications. And these are the two papers that I just showed at the beginning of the show tonight. However, mineralizable carbon has not yet been evaluated on sand-dominated putting green root zones. But remember, the more studies, both of them said even in low mineralizable soils, they should be able to detect that. And you're going to find that tonight. There's a low mineralizable soils in putting greens. The goal of this study is to determine the relationship between mineralizable carbon and creeping bentgrass growth and nitrogen uptake, which is meant to evaluate the feasibility of using rapid soil tests, mineralizable carbon determined by 1-day incubation, to predict turfgrass growth and potential nitrogen fertilizer needs. So I don't know exactly how this came about. I don't know. Part of me wants to, the cynic in me wants to go, I bet Dr. Soldat came across that paper and wanted to do it himself to see if there was any truth and did it himself. I don't know. Maybe I'm just dreaming. Maybe this just happened to be coincidental that came out after the other tests came out, but after their studies came out. But anyway, that's what's going to happen. They're going to do this test basically again on putting green soils, and we're going to see what they find. Let me read some questions. Any info from Smokey and any info on molasses? I saw it was on your BS list. Just can't find why. Oh, why is molasses on the BS list? Well, molasses is sugars, as you can imagine, and there's just not enough evidence to warrant using it. That's why. So there's exactly one paper in the literature that shows any sort of beneficial response to the application of molasses. It was blackstrap molasses from a Clemson study. And it was just one little blip on the radar. And I think it was even in the ITS journal. which is a low-tier journal. And even the author said, well, we saw something happen a little bit here, but it might just be a fluke, basically is what they said. So I just haven't seen any evidence to support using it. And if you do find evidence to support using it, you're going to have to offset the evidence that refutes using it. So, yeah, I think the concept is you're going to apply sugar to the soil, which is a carbon source for microbes, and it's going to hasten their activity or growth, which, believe it or not, that is true. That actually can happen. You can even do that just with regular mulch, just with regular, like, pine bark mulch, if it was ground up small enough. That will happen. Okay, I don't care. What I care about is turfgrass growth, turfgrass quality, performance. Is the application going to enhance that, right? And I just haven't seen enough evidence to support recommending it, so I don't recommend it. Oh yeah, that's what you said here. Yeah, I said, wouldn't that still provide some form of nutrients through, though, for the microbes, which in turn make turf healthier? So if you go back on the channel, I think if you want to look up maybe like biostimulants, or I don't remember what playlist it's under, but I went over a study that looked at applying compost relative to just St. Augustine grass. And what they found was, I think it was either done by Alan Wright or the gentleman that was at J for a while. I can't remember now. You have to go back and look at the playlist. And they were comparing the turfgrass growth and mineralizable carbon, all these other things from compost applied relative to just St. Augustine grass. And what happened is that early on, the organic carbon in the soil was really, really high from these compost treatments. And the St. Augustine grass was falling behind, right? But over 3 or 4 years, the St. Augustine grass grew in, and the root exudates from the St. Augustine grass just by itself resulted in greater soil carbon than the compost did. Because the St. Augustine grass itself, any turf grass for the most part, has so many roots and so many root exudates already. As long as it's healthy and growing, it's pulling carbon out of the air and pumping it into the soil. That's what turf grass does. It is a incredible machine at doing that. So we can go in there and artificially apply carbon here and there. And in some rare cases, if it's, really short bursts or short time spans of where you have virgin soils with nothing in it, and you need like a couple of month or so of getting it kind of primed and ready, there might be some benefit in those cases. But if you have a healthy growing turf grass, you've got carbon in the soil, and you've got plenty of carbon in that soil for microbes to flourish. As long as the turf grass is growing healthy, you've got that. So adding something more like additional microbes or microbe food or organic carbon or soluble organic carbon to do that, you may or may not find some incidental benefits here and there, but because the turf grass itself is so good at doing that already, the benefits are going to be very negligible in most cases. So that's maybe a more detailed answer than you were looking for. Alex Zamora, member, says, I put down molasses not too long ago, and I believe it made more nitrogen plant available due to the spike in microbes, and it injured my Bermuda grass, I suspect anyway. Yeah, I don't know how you put it down. If it was granular or liquid, I'm not sure. I don't know. It's hard to know really anything what happened or didn't happen there, Alex, other than you put something down and the turf apparently was injured. Was it a result of it sticking on the leaf if it was a liquid and burning it, or was it a result of some other activity in the soil? I don't know. What you're saying could very well be true. I'm just saying I wouldn't have a whole lot of confidence in what exactly caused it, other than you put this down and your Bermuda grass, unfortunately took a ding. But Joe Callis says, come on, Doc. You have to apply carbon, or why else would Site 1 have carbon Pro G? It's important to condition the soil. Yeah, I've always wondered, I don't know how, you can go back and read literature from, 1920s, around that time, 1910, 1920, 1930, and you'll see the same scams in the literature back then. It's the same exact scams. They're selling all sorts of soil wonder products, even back in the early 1900s. But the soil scientists back then, particularly with the USGA, were much more assertive with their language. And they would just call them out and say, these charlatans are just selling you a bunch of gypsum even though you don't need it. They're selling you lime even though you don't need it. They're selling you all this stuff that even though you don't need it. There's no value to it in terms of turf response. So it's never going to go away. That's where I'm going with it. These scam artists and charlatans and, all these people, they're capitalizing on our ignorance. That's what it is. We are ignorant in many regards. Don't think for a second that I don't get scammed sometimes too because I'm up here talking about turfgrass product. I have a skepticism in my mind built in that protects me quite a bit, but I get scammed too. Everybody, we're gullible. That's just the way it is. Sorry. So then they're just never going to go away. Okay. There's always going to be people out there who don't care about what's truthful. They don't have a conscience. They don't follow. They don't have the same moral and ethical compass that other people do. And they're going to take advantage of you. So that's part of what this channel is about, is to develop those critical thinking skills and to develop that a little bit of base knowledge of the literature, base knowledge of turfgrass management, in particular turf nutrient management, because that's what I'm good at. And so that you have a little bit of protection. And so the likelihood or the odds of being taken advantage of in the same regard as you might have in the past, I think the odds would be lower if you're following this channel. I'm not saying you wouldn't get taken advantage of. Like I said, I do too sometimes, but hopefully this helps build in a little bit of protection against these folks. And it's not me being cynical or not me being just ornery or whatever. It's just my experience, okay? It's my experience talking. It's just my career. I've been in too many meetings, okay? I'll just leave it at that. Too many meetings. I'll give you one anecdote. I was given a product. There were two products given to me. I'll leave the names out of it for now. But actually, I'll bring it up at some point, probably on a members only show. I'll bring up the little logo and everything. There were two products presented to me. I was a product manager at a company. And one product, both products were questionable. One product I knew didn't have any evidence to support it at all. There's nothing here that shows me that this will do anything. And the other product was like, maybe. Well, come to find out, the company was going to pick one of them one way or the other. They wanted a proprietary product to sell in-house. And there was myself, one other person like me, and then there was a salesman, the VP of sales. And we were in a meeting, us three, and he said, those two said, okay, are we okay going with this one? And I said, I'm not really that okay with it. I don't think this product does anything. And the VP of sales looked at me and said, Travis, if all we sold were products that did something, we would be out of business. And I'm like, I think I'm working for the wrong company. It's like, really? So that's the mentality. It doesn't matter whether it does something or not. It matters whether or not they can convince someone to buy it. That's what matters. And so that's my world. That's my experience. And so because of that, I have this, unfortunately, probably unhealthy skepticism when it comes to stuff like this. Okay. We continue. Sorry. All right. The materials and methods. Field experiments were conducted on 4 different putting greens from May to October 2020 and from May to September 2021 at the University of Wisconsin at the O.J. Noir Research Facility in Verona, Wisconsin. These root zones were constructed according to USGA recommendations, which requires 80% sand and 20% peat. Nowadays, it's just 20% something else. It used to be 20% peat. Now I think they amended it. The putting greens were planted with focused creeping bentgrass. Creeping bentgrass on 2 research greens were planted in 2011, and creeping bentgrass on the other 2 were planted in 2019. So we're dealing with 2 different ages of bentgrass is where he's going with that. Research greens were irrigated daily to replace 70% reference ET. Now, I mentioned this on the pro members meeting. Ninos asked me about irrigation applications, water applications. And I told them in research, we're almost always applying water based upon reference ET, and it's usually 80%. And I mentioned to Ninos in the group that was there that night, some would argue it's even a little bit lower than 80% reference ET. Reference ET being evapotranspiration of a reference crop. And in this case, the reference crop is turfgrass. And you'll see here that Dr. Soldat did exactly what you should do. And he's applying water, not every day, not 2 minutes a day, not 5 minutes every other day. He's applying water at 70% of reference evapotranspiration as estimated by an on-site weather station. Water, water, water. It's difficult. I've said it so many times. It's difficult to overestimate how important management of water is. Not applying too much and not applying too little. Apply the right amount of water. Okay. Nitrogen treatments included 0.5 or 0.1 and 2 pounds of nitrogen per 1,000 square feet every 2 weeks. This is a putting green, guys. Remember, the rates are higher. Which represents an unfertilized control medium and a high rate. Nitrogen treatments were applied 11 times during the 2020 and 9 times during 2021. Remember, this is on a putting green, so don't be shocked by these rates. These are pretty common rates on Bentgrass putting greens. Liquid urea 4600 was used for all applications. No potassium was applied. 0.5 pound of phosphorus was applied. That's it. Okay, so 3 soil cores were randomly collected within each plot and collected approximately every 5 weeks during the growing season. Soil samples were collected to 4-inch depth with a 1.5-inch diameter soil probe. Each core was separated into 0 to 5 centimeter, or so 0 to 2 inch depth, or a 2 to 4 inch depth increments, and samples representing the same depth and within the same plot were combined for analysis. Mineralizable carbon was determined by. Now this is what I was talking about. This is the reason I showed the video at the beginning. They're going to describe this, but just remember the video that I showed at the beginning. They're going to basically do the exact same thing, just a different amount, but it's the same concept. Mineralizable carbon was determined by measuring soil release carbon dioxide after 24 hours of incubation. Briefly, 10 grams of dried soil sample was placed in a 60 ml plastic cup. Deionized water was added to that, and the amount of water needed was achieved. Well, okay, blah, blah, blah. Then a plastic cup was with the soil without the cover was placed into a mason jar, just like you saw in the video. The jar was sealed and incubated in the dark for 24 hours at 25 degrees C. So it was incubated in the dark. Why? Would anybody want to venture a guess why Dr. Soldat would incubate these jars to measure carbon dioxide in the dark? Anybody want to venture a guess? The correct answer is because Dr. Soldat is an incredibly brilliant scientist. That's the answer. The reason why they do it in the dark and why we do it in the dark is because that will eliminate any potential carbon dioxide changes as a result of any photosynthetic microorganisms that exist in the soil. So the microbes are going to respire carbon dioxide, but anything in there that can photosynthesize is going to consume carbon dioxide, altering the data. This is what good scientists do. Okay? Released carbon dioxide is measured using a LICOR, which I'm going to show you right now. Okay, a LICOR is this. This is a LICOR, okay? A LICOR is an instrument. This is a handheld sort of thing, a portable LICOR, but this is, what I have on the screen is an instrument. And this little chamber thing here, this can change based upon what exactly you're trying to measure. There's all sorts of different chambers they can use, and you can all fidget with it and get it to fit your plant. But this instrument here will measure the change in carbon dioxide by running it through a series of filters and sort of gravel-looking desiccant things. That's hard to explain. They desiccate the moisture out, and then it actually measures the carbon dioxide in this box here. There's an instrument here that measures carbon dioxide through this box. This is far more precise than some little paddle, and it's far more accurate than some little paddle, and you're reading it on a colorimetric reader. It's almost like I'm watching, Wile E. Coyote trying to catch the Roadrunner or something, compared to launching the space shuttle or something. That's the difference between these two methods. Okay, this is the proper method. There's also other instruments like called gas mets that'll do it. There's all sorts of different instruments that'll do it. But this is a gold standard for measuring gas exchanges and changes in gas concentrations and so forth. All right. So that's what he used, and he corrected the carbon dioxide of an empty jar incubated under the same conditions. So what he did was, again, what you should do is that he's not just measuring the carbon dioxide in this particular jar because there's changes in the soil. He's measuring that. but then he's subtracting out or accounting for what existed in the jar just by itself with no soil. Again, it's a negative control. He's factoring things out, anything that can alter his data, like photosynthesis, like carbon dioxide changes that would exist without the soil, all sorts of stuff. He's factoring all that stuff out. So he's fine-tuning it, and so that he's trying his best to falsify his hypothesis is what he's doing. And he's factoring all the other stuff out that can fidget with it. Incubations were conducted in triplicate. Okay, turfgrass sampling. Clippings were collected from each plot approximately 3 times a week. Samples were ground into a fine powder, and then tissue nitrogen was determined by combustion. Statistical analysis. Linear regression was used to determine the relationship between mineralizable carbon and growth parameters. I don't know why I highlighted that. Okay, nitrogen fertilizer rate. Okay, so now we're getting into the results. Nitrogen fertilizer rate had no effect on mineralizable carbon regardless of root zone. Table 2. Our results are similar to studies conducted by Zhou in 1974 and Ward in 2017, which showed that additions of nitrogen fertilizer had no effect on soil mineralization of native grasslands. So if you think you're going to apply nitrogen and alter the soil mineralization, think again. It's not going to happen, probably not going to happen. And what they're talking about up here are these tables. This is the way to read it. This is over here on the left here is essentially an ANOVA. Oh, this is the ANOVA. They said ANOVA right here. And he has the depth and the nitrogen rate. And you'll see that there's no effect at all from the nitrogen. The only thing that affected it was the depth. So the top of this, the top 2 inches, were mineralizing mineralizable carbon. Yeah, it was respiring essentially more carbon in the top 2 inches than in the 2 to 4 inch depth. Pretty common result. You see that all the time. Let's go back down here. Okay. Overall, there were weak to no relationships, R squared, of 0.17 between creeping bent grass growth rate and mineralizable carbon at 2 soil depths. Similarly, there were weak positive to no relationships, 0.23, R squared of 0.23, between creeping bent grass and uptake and mineralizable carbon at 2 soil depths. Now, if you remember the other 2 articles, if you were not a member, you haven't listened to the other article. In the other article, the authors were just crowbarring their way and trying to fit that square peg into that round hole, saying, "Oh, these were highly significant. There was a relationship here when the R squareds were 0.08, 0.06. Why? Because they have a vested interest in the outcome. Now you have a paper where the authors don't care what the results are. They just want to make sure that it's accurate. And they say they were weak to no relationships, 0.17, 0.23. We'll go up here, I'll show you right here. These is clipping dry mass on the y-axis and mineralizable carbon on the x-axis. And you can see, here's the R squared, 0.04, 0.17, 0.00, 0.03, 0.08, very similar R squareds, 0.08, 0.13, 0.16, 0.11, very similar R squareds. I don't think they found any of these to be actual p-values less than 0.01 or 0.05 even. But the R squareds are very similar to the other papers, and these authors are saying, it's pretty weak. Not much there. Now, when you go down to clipping in content, and let's see, clipping in content. Oh, both these are clipping in content. Same thing, 0.09 R squared, 0.12 R squared, 0.04, 0.14, 0.15, 0.23, 0.05. Very low R squareds, meaning that the response in one variable is the variation, or the response in one variable is only accounted for by only about 5%. And when it says 0.05, only about 5% of the variation in clipping in content is accounted for by mineralizable carbon in the soil. When it says 0.17, these are different root zones. When it says 0.17, it means that 17% of the variation in clipping in content was accounted for by mineralizable carbon. That's what that means. And we just don't really have much confidence in providing recommendations if the R squared is going to be that low. I don't care if it's 0.001 p-value or not. You wouldn't say that. You'd say there's a relationship, but because only 17% of it is accounted for, we don't recommend doing this. It's too risky. You're probably not going to have a result that you're expecting, have the result you're expecting. Probably not going to happen. Okay, that's what we would normally say as scientists if you don't have a vested interest in the outcome. When you do, apparently you say it's fine, go ahead and do it. Opposite to our finding, Haney, this is the author of the Haney soil test, again a soil test I do not recommend you to use, showed a strong and positive correlation between soil mineralizable carbon and forage nitrogen uptake, which was planted on native soil. Moore, which is the paper we know I just showed at the beginning, reported a positive correlation between soil mineralizable carbon tests by using soil test kits on Kentucky bluegrass and tall fescue nitrogen uptake on a Paxton fine sandy loam, where the soil incubation was conducted at room temperature. It is worth noting that the study conducted by Moore was conducted on fine textured soils with a much wider range of soil organic matter and nitrogen fertilizer rates, with organic fertilizer and additional applications of 4 rates of urea fertilizer. Additionally, the microbial biomass, microbial activity, and soil carbon and nitrogen availability may have been substantially different from the soil used in this study, all of which can affect soil mineralizable carbon rate. This implies that soil texture or soil organic matter content and management practices would largely affect the usefulness of using mineralizable carbon to estimate potential soil nitrogen supply and plant nitrogen needs. In other words, there's so many things that are affecting it. Moisture, pH for sure. There's just texture, microsites, porosity, all this stuff's going on. Oxygen, carbon dioxide, everything's going on in the soil. There's so many things affecting it. The usefulness, they're being kind, and on the scientific literature, you kind of got to write things in a PC way. The usefulness of these tests are highly questionable given the number of variables that are influencing it. Okay? We're almost done, believe it or not. This framework is supported by the data from samples. So he's in this particular paragraph, I'm not going to read all this. It's called The Impact of Environmental Conditions on Mineralizable Carbon. He's going to talk about soil temperature and air temperature and all this other stuff in these paragraphs. You can read all this. I'm not going to read all that for you. And I cut to the end. It says, this framework is supported by data from samples collected in early May and late October 2020 and early May 2021, where the field air temperature, estimated soil temperature, and soil moisture content were significantly lower than the ones in the field. And mineralizable carbon was found to be relatively high when measured in the laboratory under ideal conditions for microbial activity. Similarly, samples collected in the summer, where the temperature is higher, often had the lowest mineralizable carbon values. While this analysis was somewhat limited by the absence of an in situ soil temperature and soil moisture measurement across the exact depth ranges from where the mineralizable carbon measurements were taken, it was clear that there were strong relationships among the environmental variables in this lab-measured mineralizable carbon, which calls into question the value and reliability of using the mineralizable carbon to make inferences about this particular system, a nitrogen-limited creeping bentgrass putting green on a sand-based root zone. Now, for those people that are managing bentgrass putting greens, if you've ever thought about doing a soil health test or ever thought about doing a Solvita test or a carbon dioxide burst test, go back and read that paragraph about another 10 times before you decide to go waste your members' money or your own money if you're a homeowner or sport turf, whatever. Basically, what he's saying is there's so much variation, don't use it. In layman's speech, that's basically what he's saying. Okay, we just don't know yet. There might be something, as I said at the beginning of the show. Tomorrow, someone might publish 10 papers and show that, yep, here's how to do it. But on April 1st, 2025, there is not enough evidence to support wasting your time and money using this particular test, the Solvita test. And I know this paper is not using Solvita, but it's using the same method and it's measuring it more accurately, more precisely by using a Lycor rather than the paddle. Okay, there's just not enough there to warrant using it. Last paragraph. In summary, although mineralizable carbon quantified by one-day soil incubation at 25 degrees centigrade after rewetting dried soil was recognized as a simple and rapid test for predicting net and soil nitrogen mineralization on native soil and estimating crop in uptake, our study indicates that mineralizable carbon is not related with creeping bentgrass growth or in uptake on sand-based putting root zones. In the specific conditions of our study, the test does not hold value for estimating creeping bentgrass nitrogen needs and does not appear to be useful for turf managers to make nitrogen fertilization decisions on sand-based putting root zones. You don't get much more assertive language than that in a scientific paper. Thanks, Doug. Appreciate that. I mean, we can't say it wouldn't work in other settings because we haven't measured it. The only papers that have come out that looked at, say, like home lawn type settings were from the people who invented it or own the technology. The two more papers. So, ask yourself, do you trust those data? And not just because the authors own it. The authors can own it and write a great paper. But because I went over that paper on Monday or a week ago, and I showed exactly why I was concerned about it, even before I realized the authors owned the technology. I didn't even know that. And it was just the way it was worded and said, and the way they said it, it just seemed strange. You don't write it that way. And it didn't, I was like, why would they say that it does work when the R squared is 0.2? It made no sense to me. Come to find out they own the technology, making money off of it. Okay. Let me read a couple questions. Oh, thanks, Alex Zamora. I appreciate your comment. Yeah. Dr. Shaddox, I'm still honing my critical thinking skills. We all are. Okay. It never ends. Okay. It's never going to end. Just keep that in mind. That's a good thing. It says, I'm still honing my critical thinking skills, not just for my lawn, but across all parts of my life. And and I'm thankful to you for doing. Okay. Well, I appreciate that. But that is why this channel exists. I've said this before, guys. I've said it before. I don't know how many times. I understand if you think this channel is about turfgrass management. I get that. But that's the instrument that I use to communicate my objective. And my objective is to hone critical thinking skills, develop critical thinking skills, or if you're just learning from the beginning, from the very start, that's fine, so that you can use them in other parts of your life other than just soils and turfgrass, right? I mean, I use it. I'm using turfgrass as a means to an end. Right. The end result is exactly Alex Zamora's comment across all parts of my life. That's one of the best comments I've received, Alex. That makes me so happy that you would recognize that, And the song at the end of the show, and there's only been one or two people have caught on to this. The songs that I play there's a reason why I play which songs I play at the end. And the song tonight is very much about that comment. Okay, just you have to read between the lines on some of the songs. Okay. Okay, guys. I'm gonna let you go. I'll be back, and it's probably gonna be like 2 or 3 weeks before I come back. It'll be a Monday morning members only show when I come back. I'll see what happens. I'm not sure. Maybe I'll pop in the middle. I come back from vacation like in the middle of a week sort of thing. So maybe I'll pop on an odd day or something, or maybe I just have to wait. But I'll let you guys know. Okay, guys. Appreciate it. Have a good little time and a break. I'll be back when I'm back. All right. Bye.